So basically, we found results for two of the four animals that we planned on testing. They were 671 and 672. We found those results, but we couldn't find results for the two samples of 608 and 609. So basically, we had to use results from two different samples to test the hypothesis. And we did samples from, it was 307 and 308. Thankfully, these samples were actually the same exact animals as before that we were using, but it was, instead of 6 months later, it was 6 months earlier. So we had it was like 6 months, 1 month, and 4.5 months, or 4.5 years, and 5 years. So basically, we could still check our results and do our data. So this is the charts of all the we looked at which of our reads were quality reads and they passed through the filters. There was also some things that were noted as unclassified, and basically the unclassified results signified that reads were not passable enough to order to be classified. They went through the first filter, but after the second level they weren't really good enough to even show that they were part of a particular classification. We did classifications at the kingdom level, order level, and the genus level. And so basically this is just the distribution of the different composition of bacteria species at the different levels. Alright, so in conclusion of a discussion of our results for 608 or 609 and 671, we found that for DNA extraction at first after the isolation, like for the very first step, we found that the isolation was successful and we didn't need a dilution in order to continue our experiment because both of the concentrations were less than or close to 50 nanograms per microliter. But for 608 and 672, we basically did too good of a job and our concentration was too high, so we had to do a dilution to bring the concentration down. We also saw that each of our samples were properly purified because the 260, 280 reading that we looked for all fell into the normal DNA range of 1.5 through 2.2. Overall, our results were good. It showed that we can move on from the first step of the purification and concentration onto the steps afterwards, and then we can do the PCR reactions. So for our PCR reactions, we looked at the gel electrophoresis, and from that it was clear that we obviously amplified our regions. All of our bars fell within the 500 to 800 range, which is good because, like I said before, we needed that 550 range. That's for DNA. And even though our negative control showed a little bit of it showed like an actual bar at the same range as all the others, it was not really a big deal because it was most likely due to like just a little bit of leakage from the ones next to it. So it really wasn't a big deal. It showed that there was a very small amount of leakage. So since these bands matched the positive control, we determined that our results were good enough to continue the experiment and move on to indexing, PCR, and cleanup. So from the fluorometric results, we found that each of our samples had a concentration above the required threshold of that we needed for our samples. There's basically you had to have a certain reading, and if the concentration wasn't good enough, you couldn't continue on with the data. Thankfully, for all of our samples, it was high enough, and for the negative control, obviously, it was too low because there was no actual DNA in there at all. So all these things showed that we were good and we could just continue going, and that our results were adequate for sequencing. So from this point on, I'm going to be talking about the samples previously collected and all these charts and data. We didn't actually have our results, so we had to use the other animals. So I'm going to be comparing the composition of animals of things collected from previous years, not actually our collected samples. So in our sequencing, each of the samples had 100% of their reads pass the quality filters, so that meant they were available to analyze. Since each of them also had 1,000 passable reads, we could adequately compare each of the samples Thankfully, for our negative control, there were only 8 reads, so it showed that there was a successful sequencing of DNA and that our results were valid. We wanted less than 1,000 for the negative control because there was supposed to be no DNA at all in the negative control. So 8 means there was like very, very little. There was just a tiny bit of contamination, so not a big deal. And the results from our positive control showed that DNA was mainly a particular species, and this species correlated to E. coli, which meant that our positive control was E. coli, and it was what we hoped for, and that the test was good. And it also proved that there was little to no cross-contamination between the samples. So, at the kingdom level, we found that overwhelming majority of the samples fell within the bacteria kingdom, which is obviously good because we were measuring bacteria. At the order level, we found that after 6 months, both animals had a greater number of bacterioids. For the dog 307-362, we saw a significant decrease in a particular order, which is known as the Clostridiols, after 6 months, and they had an increase in fusobacterials. For the dog in 308 and 671, we saw the inverse. However, the Clostridiols, the increase was only a slight increase. And I looked these particular orders up, and studies showed that the bacterials, in particular, have been associated with some metabolic diseases like type 2 diabetes. So that's going to be something to look at for the owners of the animals. So, at the genus level, we saw an increase in bacterioids, as well as Prevotella in both of the species. Along with this, both species saw an almost entirely different composition in terms of genus over 6 months. It was a huge difference in the actual composition. You can see it right here. After 6 months, each of these had a much, much different concentration and composition of their gut microbiome after 6 months. And looking at the Prevotella, they play a role in promoting increased glycogen storage. So the change in that composition obviously might have an effect on the animal after the 6 months. So we might want to look into that. Overall, our results were found to be somewhat conclusive in regards to the research hypothesis. For all of our samples compared, it was clear that there was no difference in the subjects other than that their ages were different. But after 6 months, it was clear that there was a significant difference in terms of the overall composition of their gut microbiome. So it showed that our hypothesis was somewhat correct, and it proved that after just 6 months of aging, there was a significant difference in the gut microbiome composition between the 2 animals. So overall, though, it's not really clear that it's like a direct result. There could be other aspects, such as like environment, and there could have been these were previously collected samples, so we don't actually know if all the things were correct. There could have been some outliers in the data. There could have just been difference in composition, but overall, there seems to be a difference in the composition of a species' gut microbiome as a result of 6 months of maturation. Okay, that is it. Thank you for watching.